Hello, my name is Pettinari, and today I'll be talking about bacterial range expansion and its discrepancies with the Fisher speed in nutrient-rich media. First, I would like to discuss how bacteria are able to navigate their local environment. Bacteria use two mechanisms to explore their environment: growth, which is just the cells dividing and expanding in space, and motility, which is where the bacteria are able to actively navigate their environment, usually due to flagella or other motility mechanisms such as pili. Some bacteria are also able to perform chemotaxis, where they are able to sense chemical gradients in their local environment and adjust their motility accordingly. For example, a bacterium might sense a nutrient that will help it grow and move toward that source of the nutrient. The Fisher speed is a prediction for macroscopic range expansion rates based on observed microscopic quantities such as growth rate and diffusion, which is related to bacterial motility. However, the usefulness of this prediction, as well as its applicability to real life, has not been rigorously tested in nutrient-rich media. Understanding the nature of bacterial range expansion is crucial for understanding how bacteria are able to colonize hosts, and thus is a central problem for human health. The primary question my research is attempting to answer is if the predicted Fisher range expansion speed matches the observed range expansion speed in nutrient-rich conditions. To answer this question, we used two methods of imaging. First, we used light sheet fluorescence microscopy to observe the microscopic behavior of the 6 strains of the bacteria we were interested in. This data is shown as Figure 3, with several Aeromonas tracts highlighted in pink. Second, we used macroscopic imaging with a cell phone to observe inoculated swim plates over the course of 20 hours. An example swim plate of Enterobacter can be seen in Figure 4, with time moving from left to right. As time moves forward, you can clearly see the inoculated colony expands on the swim plate. The rate of this expansion is what I am measuring for this project. The results of the image analysis performed on the data is shown in Figures 5 through 7. Figures 5 and 6 show the bacterial trajectory run speeds, which is how fast the cells were moving, and the run times, which is how long they were moving for, respectively. This data was used in combination with growth rate data to calculate the predicted Fisher range expansion speed. Figure 7 shows the observed range expansion rates obtained from intensity curve fitting of the swim plate data. Comparing the observed and predicted rates, as seen in Figure 8, we can see that the Fisher speed is dramatically under-predicting the macroscopic range expansion speed for the 6 strains examined. Looking a bit closer at the graph, we see that this black line is the ideal correlation between predicted and observed range expansion rates. For all 6 of the strains examined, they were observed to be expanding faster than predicted. This indicates that there must be some mechanism promoting range expansion in nutrient-rich conditions. Similar results were actually obtained for E. coli in 2019 by Kramer et al. The behavior that they observed was attributed to the nature of chemotaxis in nutrient-rich conditions. We plan to do further experiments to test if this is true for the species that we examined here. In the future, I plan to examine how cellular motility drives colonization of the vertebrate gut. In particular, I will be looking at if higher motility expression is an indicator of better gut colonization. Additionally, I hope to explore how zebrafish proximity affects chemotaxis of the bacteria we looked at. Since the species we examined are all native to the zebrafish gut, it would be interesting to see if they are able to perform chemotaxis towards zebrafish and uncover what specifically is inducing this chemotaxis. Lastly, I would like to thank the Partha Lab, the Gilliman Lab, and the PERS project for their continued support on the project. Thank you.